Hello everyone, my name is Sarah and I'm a bioinformatics workflow developer in the EPI2ME team. I'm going to give a brief introduction and update to EPI2ME before introducing our guest, Dr. Andre Holzer. The EPI2ME team create workflows and infrastructure to make analyzing Oxford nanopore sequencing data easy and accessible to everyone. We've created workflows which you input your base call reads into and run, and they will output reports to help you make sense of the results. We use the workflow manager Nextflow to help manage computational resources and to get you from base call reads to answers efficiently. And we use containers like Singularity and Docker to ensure reproducibility of results and to manage external packages. We now have 17 workflows which cover a range of popular bioinformatics use cases. We're always updating and improving workflows, and you can look out for release notes on our website, such as this one. One notable update from this week is the ability to process demultiplex samples as they've been specified in the sample sheet within groups. We always welcome and encourage feedback from the community, and we try to incorporate this into our releases. You can find the code for all of these workflows on GitHub, where you can also find documentation, and the analysis here represents best practice for nanopore data. You can also fork these repos and adjust them to your needs. You can find the same workflows in the EPI2ME application, which is free to download via the EPI2ME website and available for Mac, Windows, and Unix. And in terms of devices, you can run it on a laptop, desktop, a cluster, one of our sequencing devices, or in the cloud for early access users. If you're interested in becoming an early access user, then please let us know during the conference. For anyone who's used the app before, you may notice the interface looks slightly different. We're excited to introduce our new and improved interface, which is upcoming in a future release. The app opens on a dashboard, which has links to the EPI2ME website with blog posts, how-to articles, and open source data releases. Here's another screenshot of the upcoming interface. Another update is that we have now embedded IGV into the app to allow you to visualize variant calls and alignment data. Here's an example from the metagenomics workflow, which shows reads aligned with the references they've been classified from. Another exciting update is we're bringing back the Jupyter Notebook tutorials, so to provide some downstream tutorial analysis for some of our workflows. So for example, we have a Seurat tutorial coming for the output of the single-cell workflow. Another exciting update is we are partnering with tertiary analysis providers to allow access to their downstream specialist databases and analysis tools. For example, you'll be able to explore your variant files that are output by the workflows with GenX's specialist databases and analysis tools. Another exciting update is the creation of the EPI2ME file format, which will allow for distribution of workflows offline for users who cannot or don't want to access the internet while running their workflows. When developing the application, we wanted it to be a place to bring together analysis from the community. So users can import workflows that have been developed independently. To do this, there's a minimal set of requirements, which you can find on our website, and we also have a template workflow to help you get started. Some people in the community have already started using this feature, including our guest, who I'm excited to introduce, Dr. Andre Holzer, who is a senior scientist and entrepreneur who is an alumni of the University of Cambridge and Salem University, working in computational microbiology with long-read sequencing and real-time genomics. And he's going to give us a demonstration, and then we'll have some time to ask him questions at the end. So over to you, Andre.
Yeah, thank you, Sarah, for this very kind introduction, and it's a pleasure for me to be here today and present you some of our very recent research on enabling real-time analysis. So we've all heard it this morning, nanopore sequencing has revolutionized genomic sequencing, and we all love it. Yet, it brings new challenges. And one challenge is the massive data flood that many labs are actually facing. No longer sequencing is a bottleneck, but rather the analysis of the sequence data. And if you're lucky, you have someone in your lab who knows how to navigate this ocean of data. But he already knows that after data analysis, the next challenge is then to make the data available in a publishable form. And certainly, this is not what I mean. So you want to have like really nice pictures that you can easily put in your publications in the end. And even if you're lucky to have all of that, then academia hits back with its current career system, meaning that people have to move on, and you most often likely lose the ability to do what you used to do on a regular basis, meaning you have to find new people doing the job. Yet this is not what we all need. I think particularly what we need is reproducible, user-friendly, end-to-end analysis solutions to allow us to get from data to answer in real time. And this is where my vision as an independent scientist and consultant is coming together with the solution that Nanopore provides with its EPI2ME solution. And as a result of that, I would like to highlight one of the tools that we've been developing as a community over the last couple of months, which is the RT metagenomics pipeline, a real-time analysis pipeline that really does real-time analysis, and you will see that shortly. With that, I would like to give you a few examples of why we think real-time is essential. I mean, obviously, many of you might know this, but the key thing is obviously to get as fast as possible from your data to the analysis in order to make quick decision-making. And this is essential when it comes to clinical settings, but also for environmental and to reduce suffering, obviously, of people, for instance, that need diagnosed very fast. Yet, in addition, we have the possibilities of identifying the quality of the data directly, meaning we can identify abnormalities in the data, but also control when to stop sequencing. And that all comes back to the key, which is we want to be efficient as possible with our resources and ideally use what we have in the lab in best practice. And what the RT metagenomics pipeline actually provides is obviously you can use it without any bioinformatics knowledge, which is always the aim of what I'm trying to do. It's particularly designed, as I said, for real-time applications. We've optimized it for local, but also for cloud computing. You'll get data visualization right away, on the fly. We achieve that by having an interactive dashboard that provides you with this data. And it nicely, seamlessly integrates within your EPI2ME, meaning we can provide it to everyone independent of their computing solution. And this web application is also included. Yet, how did we get there? It's certainly not just been me, and I'm very fortunate to stand here as a representative of the community that we've been able to form. It's a community originating from Germany, but we're very lucky to have members and participants all over the world. And we're trying to bring people with this vision together, as we have done recently in our hackathon in Mainz, that we've been very keen to, very fortunate to organize together with Susanne Gerber's group in Mainz, as well as our collaborators here at ONT, which we're very fortunate to have. And as a result of that, we came up with a state-of-the-art best practice solution of how we as a community should be looking into development of software for long-read analysis. And this is what we came up with. So essentially, everything starts obviously with Minnow, who produces our base calls and our raw reads. And then it's directly piped into EPI2ME, where the backend is then an Xflow pipeline that is deployed via a Docker container system. And in addition, what is quite new is kind of this idea of having this front end as an additional dashboard that also allows us to directly communicate with the Nextflow pipeline in the back end. And in addition, we have some code clues that stick everything together and make it run smoothly. And with that, I would directly jump into the live demo session. Right. So here you can see the nice EPI2ME app, and as we heard earlier from Sarah, there's a ton of workflows available already that you can see under this available workflow button here. So in case you don't find the workflow that you want to use, and you have a custom workflow, as we have, for instance, you can easily install your own, and as a result here, we have our RT metagenomics pipeline installed. You then just select that pipeline, click Run this workflow. You select either it being run in the cloud or in the computer. Then the next step is obviously a very simple GUI that allows you to select all the inputs and parameters that you want to have. So obviously the key input for us is our FASTQ files. And how do we get them? Obviously we need to have some sequencing data. And let's just quickly mimic our Minnow run. So we'll get real-time base calling at the moment. So this is at the moment an empty folder, and you should see that the first sequence data is coming in. Oh yeah, so we have the first FASTQ files just streaming in. So what we'll do is select this path as our input path. So I'll just do that quickly. And then there are other options that I don't want to touch too many at the moment, but for instance, to speed up the process, I will provide a pre-downloaded database for the taxonomic classification. And for the corona, I will also provide the taxonomy here. And obviously, we want to run this in real time, so I just quickly select this here. And then for the QC, you'll have the option to get an extended QC report, but with respect to the time, we'll just get rid of that at the moment. And then we can launch the workflow. So it's as simple as that. You've started your sequencing, you launch your workflow, and you can already see it's up and running. So you'll see that the first processes are now being run in Nextflow, and you can already see here at the top, we have now 3 barcodes being base called, and you can directly see that the software recognizes these and is processing all these samples step by step now. So while this is the back end, our Nextflow pipeline, I told you about our new front end. You probably all know the reports, the static reports that we used to, that EPI2ME used to generate with the with the ONT workflows. But we've tried to bring this one step further with providing a custom dashboard, which should, can you swap back? Yeah, perfect. So this is how the dashboard looks at the moment. It says I'm waiting for some results to be produced. So they should come in every minute. Oh yeah. So you see in the back end we have the processing, and directly we get the first analysis results displayed while they're processing. So at the moment you can already see we have 2 samples being processed. We have roughly 8,000 reads, 25 megabases roughly. And you will see while sequencing is happening, this is accumulating. In addition, we have tons of interesting features, I guess all the essential features that you need for metagenomic analysis, including some QC, standard QC, but also things like diversity analysis. So for instance, here you will get nice saturation curves indicating when you've sequenced deep enough. So you can actually stop your runs at that point, as well as you get these very standardized relative abundance plots where you can directly see what are the most abundant species in the different samples. And you can, for instance, right away see that, for instance, barcode 5 and barcode 6 are different samples than barcode 8. And in addition, obviously, if this is not enough, we also provide particular targeted analysis solutions. Here, for instance, we have a potential pathogen analysis where you get the abundance of different potential pathogenic genera directly displayed. So for instance, here you already see that barcode 6 includes a high amount of reads that map to E. coli. And in addition, for instance, here now we're also showing that we can look for particular interesting species, in this case, wastewater indicators. What is coming is also a comparative analysis. We're currently working on this, as well as bringing this from the 16S analysis to the whole genome analysis, and this will also include an AMR analysis. And with that, I think I would like to go back to the slides. Just to wrap this up, we're very fortunate for the collaboration with ONT, and as a result of that, this dashboard feature will be included in some of the new updates of the EPI2ME version, so there's no longer a separate window. You'll directly have this included. And if you want to learn more, if you want to do this and use this new system for your own projects, please have a look at our poster where we've tried as a community to summarize what are the best practice ways to get there. And this is not the only these are the steps. But this is not the only example, but also we have a really nice collaboration project with Susanne's Gerber group, who has been developing this non-pro reader pipeline. And there's been 4 people that I want to highlight here, which is Anna, Stefan, Tama, and Susanna, obviously, who are also in the community today, in the audience today. So if you have any questions about how to do real-time transcriptomic analysis, they're the people to speak to. They have developed a similar system as we have, just for transcriptomic analysis that is also available and includable in EPI2ME. And as a result, I would like to sum up, again, thanking all the people that helped me on the way, and also without which this wouldn't have been possible. in particular this amazing community that is currently forming with this collaborative aspect, but also the collaborators at ONT and where everything started in Cambridge with the PanSig team, and in particular Max Stamenitz and Laura Urban, who kind of with whom I started this journey on ONT data. And with that, I'm at the end. And if you guys find this workflow helpful and want to use this in your own research, please feel free to approach me. We'll have a form to register your interest. So if you want to use that, you can get among the first to be able to access this.
Great, thank you, Andre.